Good day everyone. We are Group 1 and today we will be discussing about the first generation DNA sequencing. First generation DNA sequencing, also known as Sanger sequencing, was developed by Frederick Sanger in 1977. This groundbreaking method marked a revolutionary advancement in molecular biology and genetics, providing the first reliable technique to determine the precise sequence of nucleotides in DNA. Before delving into the intricacies of the mechanism, applications, and advantages of Sanger sequencing, it is essential to understand the historical context and significance of this innovation. The development of Sanger sequencing came at a time when scientists were striving to understand the fundamental building blocks of life. Prior to this, DNA sequencing efforts were rudimentary and limited. The introduction of a method that could accurately read the sequence of DNA was a monumental leap forward, enabling researchers to decode the genetic information encoded in DNA. This breakthrough was so significant that Frederick Sanger was awarded his second Nobel Prize in Chemistry in the year 1980 for this work, the first being for his work on the structure of proteins, particularly insulin. Sanger sequencing impact on the field of genomics cannot be overstated. It laid the foundation for a new era of genetic research, making it possible to sequence small segments of DNA with high precision. One of the most notable projects utilizing this technology was the Human Genome Project, initiated in 1990 and completed in 2003. This ambitious endeavor aimed to map the entire human genome, a feat that would have not been possible without the accuracy and reliability of Sanger sequencing. The successful completion of the Human Genome Project has since revolutionized our understanding of human genetics, disease, and evolution. At its core, Sanger sequencing involves the selective incorporation of chain-terminating dideoxynucleotides by DNA polymerase during DNA replication. These modified nucleotides lack a 3-hydroxyl group, causing the elongation of the DNA strand to terminate once they are incorporated. By generating a collection of DNA fragments of varying lengths, each terminating at a specific nucleotide, it is possible to determine the sequence of the original DNA strand. These fragments are then separated by capillary electrophoresis, a technique that allows for the precise determination of their size and consequently the sequence of the DNA. Despite the advent of next-generation sequencing technologies that offer faster and more cost-effective means of sequencing, Sanger sequencing remains a gold standard in the field. Its unparalleled accuracy makes it invaluable for applications requiring precise genetic information, such as verifying results obtained from other sequencing methods, sequencing small genomes, or conducting specific diagnostic tests.
So here are the key components and reagents that are used in the first generation of DNA sequencing. So first and foremost, you need the template DNA or the sample DNA. This serves as the original strand from which the new complementary DNA strand will be synthesized during the sequencing. Next is the polymerase or the DNA polymerase. This extends the primer by adding nucleotides that are complementary to the sample strand, creating an extended or a new DNA strand. Next would be the primers. They provide a starting point for each DNA synthesis by binding to the DNA template DNA. So they add nucleotides to the 3' at the end of each primer, hence the name primer. Next would be the D-deoxynucleotides or DDNTPs. They cause the chain termination when incorporated into the growing DNA strand, resort, resulting in fragments of various lengths that end at each possible nucleotide position. These fragments can be used to determine the DNA sequence, so they cut or they stop the sequences. So next would be buffer. So this provides the necessary ionic environment and pH level for the optimal activity in which the DNA polymerase wants to during the sequencing reaction. Next is the sequencing gel or capillary electrophoresis system. This is like the labeling system of this sequencing. So this separates the DNA fragments based on their sizes. Short fragments move faster, while the longer fragments moves slower through the gel or capillary, allowing for the determination of the sequence. Next would be the detection system. As the name suggests, the detection records the labeled fragments as they pass through the gel or capillary. This allows the sequencing to be read. Lastly, the thermal cycler, or more commonly known as the PCR machine, used for thermal cycling during the sequencing reaction to repeatedly denature the double-stranded DNA, anneal primers, and extend the new DNA strand. This ensures multiple cycles of DNA synthesis and termination, producing a complete set of terminated fragments for sequencing.
The Sanger sequencing, or chain termination method, has three major steps in its overall procedure. The first step involves the integration of fluorescent chain terminating DDNTPs to the polymerase chain reaction procedure. Here, the standard PCR process is executed, which involves the selection, isolation, and amplification of a specific DNA segment. The prime modification in the said method is the addition of certain modified nucleotides or DNTPs called dideoxyribonucleotides or DDNTPs or dideoxynucleotide triphosphates or DDNPTs. They are typically altered to be fluorescent and serve as the agents for determination of elongation. During the elongation step of PCR, DNTPs are added to the growing DNA strand using DNA polymerase through catalyzing formation of phosphodiester bonds between the last and succeeding nucleotide. A low portion of chain terminating DDNTPs is integrated in the PCR, which allows the DNA polymerase to randomly incorporate the said nucleotides during the elongation step. Since the DDNTPs lack the needed hydroxyl group for phosphodiester bond formation, the elongation process is terminated once the nucleotides are added. Overall, the modified PCR process results to millions to billions of oligonucleotide DNA copies of the selected segment, terminated at random lengths. The second step involves the size separation through capillary gel electrophoresis. Here, the chain-terminated oligonucleotides are separated based on size through gel electrophoresis. The samples are loaded into one end of the gel matrix, and then an electric current is applied. Since DNA is negatively charged, the oligonucleotides will be pulled toward the positively charged electrode at the opposite side of the gel. The DNA fragments exhibit a constant charge per unit mass, and thus the speed at which the oligonucleotides move through the gel are determined solely by their size. Basically, their size is inversely proportional to their speed. A resulting arrangement of oligonucleotides, arranged from smallest to largest, read from bottom to the top of the gel, is generated by the process. The third step involves the application of sequencing machine. So the final step is simply reading the gel to determine the sequence of the input DNA. Since each terminal DDNTP corresponds to certain nucleotides in the original DNA sequence, the sequence can be read based on the determined sizes. For example, when it is determined that the shortest fragment terminates at the first nucleotide from the 5' and the second shortest fragment terminates at the second nucleotide from the 5' the original DNA 5 to 3 sequence can be determined by reading the gel bands from smallest to largest.
First generation DNA sequencing is widely recognized for its precision and reliability in validating genetic sequences, standing as the gold standard in DNA sequencing. With the capability to produce lengthy read lengths of up to 1,000 base pairs, this method excels in accurately sequencing specific genes or small genomes. Having been extensively used since the 1970s, it boasts a wealth of established protocols, knowledge, and tools, rendering it well understood and easily accessible. However, first generation DNA sequencing does have its limitations. It falls short in throughput when compared to next generation sequencing technologies, making it less suitable for large scale genomic projects. Additionally, the process is labor intensive and time consuming, demanding significant hands on time and preparation. Moreover, the cost per base pair is higher for large genomes compared to NGS methods, rendering it less economical for extensive sequencing projects.
Now we will be discussing the applications of first generation DNA sequencing. This has been an underlying basis for advancing our understanding of genetic medicine and other fields. So let's dive into the different areas where first generation DNA sequencing has significantly contributed. So first we have genetic research and molecular biology. First generation sequencing has been pivotal in genetic research and molecular biology. It allows for identifying and characterizing genes, helping us understand their structure and function. Researchers can also perform mutational analysis, identifying mutations that cause diseases. It is crucial in evolutionary studies, allowing scientists to trace the evolutionary history of organisms by comparing their DNA sequences. Another application is medical diagnostics. This has paved the way for personalized medicine where treatments are tailored based on an individual's genetic makeup. Furthermore, carrier screening using this sequence to identify individuals with genetic mutations that could be passed to their offspring, which is especially important for recessive disorders. Third application is forensic science. Forensic science is another field that has greatly benefited from the sequencing. So DNA profiling is a crucial application that is used for identification in criminal investigations and paternity testing. It can also help in ancestry and lineage tracing, providing insights into generic heritage and familial relationships. This technology has been crucial in solving crimes and identifying individuals in various forensic cases. Fourth application is biotechnology and pharmaceutical development. First generation sequencing is employed in gene cloning and expression studies in biotechnology. It is essential for producing proteins and other biomolecules. Of course, it is also instrumental in drug development and target identification, helping pharmaceutical companies identify genetic targets and understand drug mechanism. This has led to a more effective and efficient treatment for personalized therapeutic strategies. Last but not the least, we have agricultural and environmental sciences. First generation sequencing has applications in agricultural and environmental sciences as well, as it aids in crop and livestock improvement by identifying genetic markers linked to desirable traits, thus supporting selective breeding programs. biodiversity and conservation genetics, it helps assess genetic diversity, which is critical for conservation efforts. Moreover, in microbial ecology, it helps characterize microbial communities, providing insights into their roles in various ecosystems. So these are the applications which demonstrate the broad use of first generation DNA sequencing in advancing scientific knowledge and improving practical fields.